Go ahead.
Yeah, thanks. I will keep it straight to the point. My sincere apologies, but it's my great pleasure again to be here presenting my humble work, L2 Unifrac, finding phylogeny-aware and biologically meaningful averages of metagenomic samples. And this is a brief outline of the contents. First, I'll talk about the background and motivation leading to this work, and the method and principles that for this method to work, and the problem we faced and the solution we proposed, and some of the evaluations that prove the effectiveness of our method. And let's begin with the background and motivation of this work. So microbiomes are everywhere. It can be in the soils, in the sea, and on our bodies. And they also tell us about many things, like the pollution level of an environment or the health status of their person. But there's also challenge is that much evidence has shown that we can distinguish distinct environments by the microbial composition, but it's really hard to really describe a specific environment by its metagenomic composition, in the sense that it's hard to say how to point at an environment and say, oh, this is what a typical sample from this environment would look like, high in this species and low in that, and things like that, right? So here's an example. We have two independent studies of the same condition of colorectal cancer and its correlation to gut microbiome. We can see the same similar species being picked out, such as firmicutes and bacteroidetes, that they believe have differential abundances in healthy samples and in diseased samples, but the trend that they reviewed are kind of different, right? For example, in the study in the left, in this picture in the left, we can see it shows that the firmicutes decreases as their cancer deteriorates, whereas on the right we can see that the worst condition actually has the highest firmicutes, but whereas for bacteroidetes, we can see that they captured a similar kind of trend, that healthy samples have significantly less bacteroidetes, right? So this really makes us wonder that if we can distinguish their environments through metagenomic composition, then is it possible not only do we distinguish them, but also describe them and giving each environment a unique signature, right? So this is how we thought about doing it. So what initially led us to believe that such thing can be done is the fact that samples, metagenomic samples from different environments cluster. For example, in this picture, we see that the samples from different body sites roughly clustered together. Right? And here we also introduce a concept called barycenter, which is we can roughly see as the center of the cluster, but mathematically it represents a point that minimizes the sum of distances to all the other points, right? So if we are to represent one cluster with one simple sample, one single point, then the most natural choice would perhaps be the center of that point. Then the question would be then what distance, because depending on the distance metric, given the same coordinate systems, but depending on the distance metric, the clusters may look different, and so with the barycenter, right? Then which distance metric is suitable? So we propose using Unifrac. The reason being that it's a widely used metric, and it has been shown to be robust in many metagenomic studies. And also, very importantly, it has a very nice mathematical characterization that makes it very easy to compute. And we'll talk about it in the next few slides. And here we talk about the setup for Unifrac computation. So the computation of Unifrac by its original definition is kind of complicated, but there's a alternative formulation. So under this formulation, suppose we need to compute the Unifrac distance between sample 1 and 2. We need to have a phylogenetic tree with that includes all the species, all the organisms in these two samples as nodes on the tree. And we also need to have distances, branch lengths that each representing the evolutionary distance between the two nodes of branch is connecting, right? So, and then a sample is then represented as a vector indexed by the nodes in the tree. And it's also in the form of a probability distribution in the sense that each entry represents the relative abundance of the organism in that sample. And in this way, each entry, each term of the vector is non-negative, and all the terms should sum to 1. So under this setup, this is how we will compute the unifrac between sample 1 and 2. It's a two-step process, and the first step we call it aggregation. So we use this example and show a visualization of it. So first we have the relative abundances of the samples assigned to its to the corresponding positions on the tree. And then starting from the lower parts of the tree, we will start aggregating the messes up the tree, multiplying by the branch length on the way and adding it to the ancestor as shown in this simple illustration. Yeah, so in this process, we get a new vector, which we will call the aggregated vector because it's obtained through this process of aggregation. And we will do the same for the other sample and obtain another aggregated vector. And the second step is to compute the L1 norm of the difference. So we subtract the two vectors and we compute its L1 norm, and this computes the exact unifrac distance. And these two steps can be combined into one line with the aggregation process represented by a invertible matrix, which we call W here. And we call this formulation of unifrac computation the L1 unifrac because it's computed as an L1 norm, right? So how would this help us find the barycenter with respect to unifrac distance? So we can look at this aggregation process in this way, in the sense that the samples are viewed as vectors lying in the distribution space, and through aggregation, their points are sort of projected into a different space, which we call the L1 unifrac space, because when we take the L1 distance in this space, it computes the unifrac distance, right? So in a way, each of the points in the distribution space has a unique correspondence in the L1 unifrac space, which is its aggregated form. And an interesting thing here is that since W is invertible, this process is also reversible. And we call this process the reverse process of this, the reverse aggregation, which is simply multiplying by the inverse of W. Right, so let's consider this intuition. Suppose I have a bunch of samples in the distribution space, and I aggregate all of them to the L1 unifrac space, obtaining a bunch of aggregated vectors. And then I find the barycenter with respect to L1 distance in the sample, which is simply the median of all the aggregated vectors. And what if I try to reverse this process? Right? Will I get the point in the distribution space, which is the barycenter with respect to L1 unifrac, and one that we can use as a representative of all these orbital samples, right? Unfortunately, the answer to this question is no, because we run into a huge problem of negative values. So in this picture, we did an experiment. We gathered a bunch of metagenomic samples from different body sites and computed computed the supposed barycenter according to the method we just described. And this column of numbers we see here represents the number of negative values in the vector that's obtained in this process, right? Then why is this a huge problem? It's because it means that if we have negative values in a vector, it deviates from our purpose of wanting to find a representative sample that is biologically meaningful. Because after all, what does it mean we have negative abundance of certain species in a sample, right? It doesn't really make biological sense. And why is this happening? What actually went wrong? Right, the problem lies in the taking of L1 norm is because probability, mathematically, probability simplex is not closed under L1 averages, which means that when I take the L1 average, which is the median, in the L1 unifrac space, and when I try to reverse aggregate it, I might not get a point that lies in the distribution space. It might be somewhere like outside of it, right? Then what is their solution? Right, we proposed a very simple fix. We proposed a new alternative metric called L2 unifrac. Basically, instead of taking the L1 norm in the aggregated space, we will now take the L2 norm instead. So as such, the L1 unifrac space will now be called the L2 unifrac space. And instead of taking L1 average, which is the median, we will now take the L2 average, which is the mean. Right? And we claim that in this way, we will obtain, when we reverse aggregate it back to back, we will actually get the point that lies in the distribution space, and that is the barycenter with respect to L2 unifrac. So before moving to proving this claim, I would first like to show you the similarity between L1 unifrac and L2 unifrac, because we originally chose L1 unifrac because it's robust, right? But what if L2 unifrac is not? It's kind of meaningless. So we gathered 528 samples, and we computed pairwise L1 unifrac distances and pairwise L2 unifrac distances and computed the correlation coefficient between them. And we can see that they have there is a high correlation between these two kinds of scores. And this means that L2 unifrac actually captures the same essence of L1 unifrac in terms of measuring metagenomic dissimilarity, right? Then we did some mathematical work, and we proved that the beauty of L2 unifrac is like we will not get negative values, and the resulting vectors will always be a distribution. And this is the proof, which I will not go into detail. And secondly, this process of aggregating and taking the average and reverse aggregating, it computes the barycenter with respect to L2 unifrac. And this means that if I were to cluster a bunch of samples using L2 unifrac as the distance metric, the vector that I found in the method that I previously described is indeed the center of this cluster, right? So combined with the previous result, this shows us that we have indeed find a way to compute a biologically meaningful representative of a group of metagenomic samples. And number three is a very interesting result. So we actually show that this vector obtained through first aggregating and taking the average and reverse aggregating is the same as taking the component-wise mean of the original distribution. Yeah, which means we can further simplify and speed up the process. Yeah, and this is not something trivial because L1 unifrac doesn't possess this very beautiful property, right? But for L2 unifrac, this is possible, which makes life even easier. And next, I will talk about some applications through which we can understand the beauty and the usage of average samples computed with respect to L2 unifrac. So first, let's look at this picture of how average sample with respect to L2 unifrac will look like with respect to the other vectors. We can see through this PCOA plot that is indeed the center of the cluster as shown by this orange points. And this means that we can start fingerprinting environments by representing an environment using one single vector, right? Then what are some of the applications. And first, we did the classification test to see the usefulness of this method. And in this test, we used again the metagenomic samples from different body sites. And for each of the body sites, we used 80% of the sample to compute the representative sample using the method we described. And for the remaining 20% of the samples, we will compute the L2 unified distance to each of the representative samples, and we will assign a body site to one that's closest to this sample. And we measure the accuracy of this method, and we can see it's pretty high. And in contrast, we compared it to some of their traditional methods. So traditionally, classification is usually done through clustering because there wasn't any kind of reference through which we can classify, right? So usually we have to cluster the samples first, and then depending on the cluster, we will decide the really the label of certain sample, and that's how classification was usually done. And we compared it to two kinds of clustering methods, k-means and k-midways, and we can see that the accuracy score is significantly lower than that using a reference point. Right? And another usage of this representative sample is that it makes comparison between environments easier, because now we only need to look at the comparison between two samples instead of two large groups of samples, right? So the plot shows an example of it, showing a comparison between the representative cancer samples and that of control samples, and their differentially abundant organism that's reviewed through this process. Of course, I believe that much more analysis was needed to really confirm the effectiveness of this method, but here we present a method that I believe has a lot of potential to it. And lastly, the presence of L2 unified space actually gives us an alternative way of clustering metagenomic samples. Right? So originally, if we want to cluster samples with respect to the unified distance, for example, we'll first need to compute pairwise unified distances. Right? And based on that pairwise distance matrix, and clustering is performed. And in this process, we can see for each sample, aggregation is done n times if we have n samples roughly, right? Because for any pair of computation of unifrac, you need to aggregate them. And we propose the alternative way of clustering the samples by first aggregating all of the samples to the L2 unified space and clustering them using L2 norm as a distance metric. And what will happen? So we see that there's indeed a slight decrease of clustering quality, but in exchange, there is a huge improvement in efficiency. And it's kind of expected because the original method, each sample is aggregated. You perform aggregation n times for each sample, but for this, for our method, you only do it once, right? So it's much faster, and the decrease in quality is really very. I believe it's kind of insignificant, and this means that it provides an alternative way if we want like a clustering result in a very short amount of time, and which we would tolerate a little sacrificing quality. And just as a side note, that this method also works if we perform the L1 unifrac, and it might probably has better result than L2 unifrac. The exact reason for this difference in clustering quality, I do not quite know, and I think it can be quite an interesting mathematical problems for those who are interested. And as the conclusion, we proposed a alternative distance metric called L2 unifrac, which works similarly as the traditional L1 unifrac in terms of measuring metagenomic dissimilarity, but it has additional advantages that cannot be achieved with the traditional unifrac. And namely, it allows us to compute biologically meaningful representative samples. And also, in addition, we introduced clustering in the L2 unifrac space, which allows highly efficient clustering of metagenomic samples with negligible compromising quality. And that's the end of their presentation. There's a link to the manuscript for those who be interested, and there will be a code for a prototype of their um, method we implemented. And lastly, I would like to acknowledge my lab, especially Dr. Flicky, who gave invaluable guidance to this work, and also Andrew did quality for this work, and last but not least, NIH, who funded this work. And thank you.
Thank you, Wei, for giving your talk. I'm sorry about the technical difficulties. So, does anybody have any questions for the speaker? I was wondering.
Yeah, go ahead.
Okay, I was just wondering whether you've made this available as Python or something.
It's in Python, but it's a prototype.
It's what? Sorry.
We have a prototype. It's written in Python.
Okay.
And
okay.
Yeah, we included a link in the manuscript, and I will also be happy to share with you the code.
Okay, thank you. There's another question.
Hi, this is Christian. Thanks for the nice talk. Have you tried to do log ratios instead of your relative abundances? Because when you aggregate over the logs of your composition, you get geometric means, which are more meaningful for your data than averages over the aggregation.
Is that for the purpose of getting rid of negatives, or what would be the purpose of it?
It's a different type of averaging.
Uh-huh.
It's a different type of averaging which has been used before. So for instance, if you did prediction right away, you would use a log contrast model where you could aggregate over geometric means of subtrees and not averages of what you have now.
And I haven't, but I think it's interesting to try different kinds of averages. We thought about too, just because it's a natural process, natural advancement from one norm, but we did think about as direction to think about other forms of averages, such as LP norm in general, to see whether they're nice mathematical properties that they might have. And another reason, which I do not know much in detail, is that L2 unifrac does produce some kind of number that it actually has some kind of biological meaning that somebody has studied before. Yeah, so, but definitely we wanted to try more ways of averaging.